this. What I will do during the first, I would say, 20, 25 minutes, I'm going to provide you a journey through my 6 years of skeletal muscle research. That's where my initial basic science has been for the last 15 years, until I changed dramatically my laboratory in January, and I will tell you about what I have changed to. I guess I have to make the disclosure that I'm a scientific advisory board member to a company called Solid GT that is involved in Duchenne muscular dystrophy gene therapy, and Mitochine, that's a company that's involved in identifying mitochondrial compounds that improve mitochondrial function. So when you look at muscle and muscle diseases, there are a number of inherited and acquired forms of muscle diseases that can lead to loss of muscle mass. So this is a patient of mine who has, despite all exome genome sequencing, is still unidentified congenital muscular dystrophy. I don't know what she has. This is a boy who has Duchenne muscular dystrophy. That's a girl who has a milder form of congenital myopathy. And this is a boy who simply has a de novo duplication of chromosome 4p, which we assume to be disease-causing. Although I don't really know how that duplication relates to his significant muscle weakness, which he has. But again, it's an inherited form of muscle disease, as opposed to the acquired one. So who in this audience had a cast once in their life? So you remember what happened when you took out that cast, right? So you develop quite a little bit of atrophy when you go through disuse conditions. I'm not sure whether anybody has made it up to space, but if you come back from space, one of the major problems in getting adjusted to Earth life is bone and skeletal muscle weakness. That's the biggest issue of people when you don't have gravity, and here you have significant muscle loss in patients with cancer or HIV. But one of the biggest issues which we all face, and I have to tell you, anybody in the audience, after the age of 50, 1 to 2% of your muscle is lost over time, no matter how much you exercise. You just preserve whatever is still there better when you exercise, but you lose it still. There's sarcopenia, age-related muscle loss, and there's about $26 billion healthcare costs in the United States that just result from sarcopenia. So muscle diseases span the entire spectrum from rare to common diseases. And what I'm going to do is I'm going to walk you through an example of two different genetic disorders that has really kept my laboratory alive for the last 6 months. And I'm going to start with Marfan syndrome, which is a connective tissue disorder. How many non-clinicians are here? Okay, good. And I will take time to, and I didn't want to bore you with background, you know. So Marfan syndrome is a connective tissue disease that affects many different aspects of your body, your eye, your joints, your spine, but most importantly it affects your aorta because you're at risk of aortic aneurysm, and if that aneurysm dissects and ruptures, then you have a major clinical problem which can lead to death. And I became interested in Marfan syndrome one day when Hal Dietz, who is, I think, the maven of Marfan syndrome at Hopkins, who was my mentor and attending, called me one day to clinic to see Quinton. Quinton is a boy who has a severe form of Marfan syndrome, but as you can see, he sits in his chair and can't walk. And Hal told me, I think there's a muscle component to Marfan syndrome, you need to study this. And I never was really that interested until I saw this boy. I'm like, oh, there's a muscle component to Marfan syndrome, I need to study this. And interestingly enough, this is the original patient which was described by Professor Marfan. And look at his muscle. I mean, there really is not much muscle mass there. And then I figured, okay, and then I saw another baby that was born in the NICU who was incredibly weak, had Marfan syndrome. But when you think about Marfan syndrome patients from a clinical point of view, or even the non-clinicians, you hear about basketball players, so there's not much about weakness. But what was interesting is I started to then interrogate the families and ask them questions. And although they're really not allowed to do strenuous exercise, some of them still do, and they were always frustrated they couldn't build up muscle. I always said, I know I'm violating the rule, and then it has no turnout. And that's where I got interested. And I'll show you why in a little bit. The other disease I will talk about is Duchenne muscular dystrophy, which is the most common form of muscular dystrophy described by Professor Duchenne. There are some arguments who described this first. It's still Duchenne muscular dystrophy. The person who probably really described this first was Edward Marion, who, I have to tell you, came up with the idea of why is muscular dystrophy happening in a paper which was published in 1852. So sometimes you see the great minds. He didn't have PCR or he didn't have anything, but he knew what this disease was all about, because he said the sarcolemma of the muscle fiber is broken down, is destroyed, and that what makes these muscles weak. And he was right, because 130 years later we ended up here, where dystrophin as a major protein of the sarcolemma was discovered, which is connected to lots of other proteins. They're all called the dystrophin glycoprotein complex. And that connection is lost in Duchenne muscular dystrophy. Because here you have a normal muscle that shows you dystrophin staining, and in Duchenne muscular dystrophy you have none of it. And I'm going to show you now how you can get insights into the pathophysiology for muscular dystrophy by studying patients with Marfan syndrome. So when Hal showed me this patient and I convinced myself there's something there, I was lucky enough that there was an orthopedic surgeon in Hopkins who, every time he did surgery on the spine of Marfan syndrome patients, he took out a piece of muscle because he thought, well, maybe someday somebody will be interested in this. And that someday was when I knocked at his door and said, I want to have these samples. And so first of all, you can see that Marfan syndrome patients have no fibrillin 1. That's not unexpected. You would expect that. Then when you look at the muscle, so this is how normal muscle looks like under the microscope. Pretty much lots of little stop signs all close together, all the same size. And here you see lots of tiny fibers. Then you see fibrotic tissue here. It was a really abnormal muscle. And then Hal made a mouse model for Marfan syndrome. We studied the mouse, and we saw similar abnormalities in the muscle. So much of the work of Hal was really to show that an excess of TGF-beta signaling is the real cause of Marfan syndrome. So for years he was thinking fibrillin 1, which encodes for a gene that's really forming the structure of all sorts of tissue, connective tissue, is a structural disease. And it frustrated him because fixing a structural disease is difficult. But 15 years after he discovered the gene, he figured out that it's actually a signaling disease, because excess TGF-beta signaling leads to a lot of downstream parameters that have an effect on the disease pathogenesis. So what we had to do is we wanted to see whether TGF-beta signaling plays a role in the muscle of Marfan syndrome mice. And how we did this is we looked at SMAD2/3 and periostin staining, and you can see there's a lot of staining here for both of those, pretty much as an indicator to tell you there's too much TGF-beta signaling. How do you prove this as a cause of the muscle abnormality? We treat them with a TGF-beta neutralizing antibody, and you see that the TGF-beta signaling disappears, but also the muscle structure turns really close to wild type. So here you establish the causative relationship between a signaling abnormality causing a phenotype. One of the interesting things, and now we're coming back a little bit to the people who are going to the gym, who should be going to the gym, is that we saw what we call in muscle split fibers. So you look at this, and these split fibers usually indicate that the muscle doesn't repair really right. So the muscle is trying to repair, but it doesn't do it quite right. So everyone in this room probably had muscle aches at some point, I would assume. Not everybody's nodding. I hope everybody in this room had a flu shot. Best people are nodding, it's bad. So every time you get a flu shot, you destroy about 3,000 muscle cells. But in patients who repair this route, it doesn't matter because it's just being repaired. Because skeletal muscle is a highly regenerative tissue as opposed to other tissues like the brain, for example. Because you have these quiescent progenitor cells that are called satellite cells. They get activated, they go to the damaged fiber, and pretty much in the process of probably a couple of weeks repair all this damage. Now you can study this damage in mice by injecting a snake venom called cardiotoxin. So you take this into your little syringe, you inject it into the muscle, and then you cause a lot of damage to the muscle, and you try to see how well is a muscle repairing and regenerating. And if you look at fibrillin Marfan syndrome mice, you see that a wild type mouse shows a lot of these round cells, they're all regenerating. None of that can be seen in the fibrillin mice, and if you give them TGF-beta neutralizing antibody, you restore that regenerative capacity at early days and as well as in 18 days, where they then repair with this. So not only that there's a structural abnormality to the muscle, it really is the repair muscle that doesn't work, and you need to have this repair mechanism in place to build muscles. So if I decide I want to look like Arnold Schwarzenegger, then I need this mechanism to be in place to build my muscles, and that's why most of Marfan syndrome patients, although they don't really have a lot of weakness when they get older, they are still very thin. They don't have much muscles because they can't build it. So how is that related to Duchenne muscular dystrophy? I showed you this picture before, and I told you that if you lose the dystrophin protein, the entire complex is disrupted. And when I did my postdoc 13 years ago, I think, no, 15 years ago, sorry, I went to the laboratory of Dr. Kevin Campbell, who is the muscle maven, in my mind at least, one of the ones. And what we thought about the pathogenesis, what I told you already from the 1852 paper, there's no dystrophin, you lose all the complex, and you lose the linkage between the cytoskeleton and the basement membrane. And then you enter the cycle of cell necrosis, regeneration, and at some point that cycle is disrupted. Because for the clinicians here, you know that, but for the non-clinicians, when you see a boy with Duchenne muscular dystrophy, you can't pick them out initially. They're walking clumsy and not 100% normal, but they are walking initially. Just over time, as the disease progresses, they become weaker and weaker and weaker and end up in a wheelchair eventually. And that's when the circle is kind of disrupted, and instead of making new muscle, they're making scar tissue, fibrotic fat tissue, and that's when they become weak. And one of my contributions to the Campbell lab in Iowa City was that satellite cell function, so these repair muscle cells, decrease with age in muscular dystrophy, and that leads to severe dystrophy and fat fibrotic tissue. And then when I started to work in Helen's lab, I wanted to test the hypothesis whether increased TGF-beta signaling, as I showed in Marfan syndrome mice, has an impact on satellite cells in muscular dystrophy, as well as we know, has an impact on scar tissue formation. So how did we test this? We did the same assay I showed you before. We looked at MDX mice that have increased TGF-beta signaling markers, which was kind of exciting to see, but we obviously had to go through the same process of proving causality here. So here we gave TGF-beta neutralizing antibody to the MDX mice, and we restored regeneration. It's just a different assay. All the red cells are regenerating cells. You see that this MDX, the dystrophin negative mouse model, has no regeneration, and then if you give the neutralizing antibody, you restore this. Now that's all good and nice, but who wants to give a patient a neutralizing antibody which you buy off the shelf from Sigma? Also can't do that. But one of the early work from Hal's lab was that losartan is a blood pressure medication that's been taken by roughly 5 million people around the world, FDA approved, not expensive, does not have any real side effects, to be honest. I know it's the non-clinicians, or some of the clinicians may not agree with me. Every drug has a side effect, but that has a very low side effect profile. And it's also known to inhibit TGF-beta signaling. So we took our muscular dystrophy mice and we gave them losartan. We repeated pretty much the same experiment and restored the entire regeneration machinery with losartan the same way we did it with the neutralizing antibody. And then obviously we wanted to see whether it has an impact on the lifelong disease of the mouse. So we treated the mouse for 8 months, and you can see that the losartan-treated mice had significantly healthier looking muscle than the non-treated mice. They were also stronger when you looked at the strength. And so we had some very successful results from that. So if you take this together, we could show from one disease that TGF-beta signaling plays a role in the muscle in that disease, and it also plays a role in the other disease in muscular dystrophy. And these were really data who provided grounds for a clinical trial to be conducted in Duchenne muscular dystrophy, which I have to say, this work is now several years old, and the person who I charged to run this clinical trial still hasn't gotten his act together. So somebody else is going to do it now, which is good. But by now, what happens in clinical real life, if you have a drug that's not expensive, that doesn't have any side effects, parents call you and say, can you give it to me anyway? And what do I say to the physician? No, we need to wait for a clinical trial, but come on. And then I prescribe it. How can I know? We'll have that discussion at the end of my talk. I want to go back to Marfan syndrome and losartan just quickly to tell you why it's kind of nice that it does something to the muscle, but most importantly, it rescues the aortic phenotype of the Marfan syndrome mice. And I want to go back to Quinton, who we started on losartan at this stage because he was so young that we wanted to avoid surgery of the aorta at all costs. Because if you do surgery below the age of 10 or even 15, you're pretty much guaranteed to have to do surgery again. We want to avoid that, of course. So we did compassionate use of losartan therapy. And his aorta stopped growing, but from the mother's perspective, he turned into a normal boy who all of a sudden could walk and run within 18 months. So there's a multidisciplinary, multi-center clinical trial for losartan and Marfan syndrome, which has just come to a conclusion that's going to show that losartan will be the new standard of care treatment for patients with, the connection is lost to the meeting. Should I try to reconnect? Sorry. But there's an ancillary study that looks at the muscle phenotype as well. It will be interesting to see whether muscle mass is also going to increase in these patients. And now I'm going to show you a completely different area of research. So there are 3 different areas of research I'm going to talk about. That's the second. And I'll tell you how that came in my heart. Because when I started at Hopkins, I was working on this losartan stuff, but that was all published kind of by the time it started. And when I wanted to apply for a big NIH grant, you know how it is, similar to CIHR, you pretty much have to have the thing already done by the time you apply for this, and I didn't have much done. So there was an Innovator Award given out by the NIH for just sexy ideas. No preliminary data needed, just sexy ideas. So I started to become interested in hibernation, and as cheesy as it sounds, bears are my favorite animals. And I was always fascinated as a kid, and upset as a kid when I went to the zoos in Germany, where I grew up. It's not that cold. You can go to the zoo all year long. But in the winter, obviously, you don't see the bears because they're hibernating. Now, you will imagine that using the bear as a model for your lab research is not very practical. My wife wasn't happy about that. But then I had found this. Believe it or not, sometimes life gives you good stories. While I was writing this grant, that animal was in my backyard. You know what that is? It's a woodchuck. That's also a hibernating animal, which I have to admit I never paid attention to. And there's actually a company in North Dakota in the United States that offers scientists access to hibernating woodchucks because they don't get hepatitis. Now that's not really what I'm interested in. So I found these animals. And these are hibernating ground squirrels, 13-line ground squirrels. And I found a wonderful lady through Google is the best thing ever. I found her through Googling. A lady who sits in Oshkosh, Wisconsin, has a colony of hibernating ground squirrels because she's interested in retina research. She does retina research in hibernating animals. And when I told her about my idea, she was just fascinated. She said, sure, I collaborate with you. And that's how that started. So if you look at animals during the fall, this is what you do. There's no Trader Joe's in Canada, so I don't know what, you don't know what that is. So I don't know what food store is in equivalent. It's like a health food store where I had to go once a year and buy about $200 worth of unsalted natural nuts. And you feed these animals during the winter, and then they take on this ball-like structure because they build all this fat. And then during November, December, they take on what's called the torpid fetal position, and they go into hibernation. And then they go to sleep. In a laboratory setting, you put them in a refrigerator. Yeah, that's actually, that's what you do. And fully approved by the animal care. Also in Toronto now. I bring them to Toronto now. I got a grant to do this. And the only thing they do every 2 or 3 weeks, so they don't eat, they don't drink, but every 2 to 3 weeks, they wake up. And what they do is, at this stage, their heart rate is 3 beats a minute. They breathe 3 times per minute. So it's like shut up. But then they wake up, heart rate goes back to normal, and the only thing they do is they urinate. Which I don't know what it means, but I think it means something really interesting. And I had a guy at Hopkins who was a nephrologist, but he was 78. He started to do some research, and then he said, I have to retire now. That's enough for me. And then they wake up in the spring, and I show you just a couple of videos how that looks like. So that's a hibernating squirrel. And they are quite resistant. You can do a lot of things with hibernating sleeping squirrels. They don't mind. Otherwise, as cute as they look like, nasty animals. And this is a squirrel that just woke up. So I want you to just look at this for 2 seconds. So that's after 6 months of not eating, not drinking, and most importantly, not moving. So obviously, these animals have figured out what thousands of researchers trying to figure out: how do you protect muscle mass? That's what I got the grant for. So we started to first look at the muscle, and as a muscle biologist, I have to tell you, I was surprised that this muscle literally looked normal. I was sure that it was something abnormal. I was thinking that it would store something there, there would be fat there. It looks really indistinguishable. And then if you think about how do you maintain muscle mass, it's really the homeostasis of making enough protein, not breaking down too much protein, and muscle regeneration, as well as mitochondria that play a role in muscle maintenance. And I will tell you honestly, I got my grant for this because I thought satellite cells are the driver of hibernating and protecting muscle, and I think people gave me the money because they thought it's so cool to study the stem cell in hibernation. Yeah, it's not. So you can kill satellite cells by irradiating them. You get rid of like 95%. That's what we did. I'm not going to show you all the data. We did a whole study of this. You irradiate the muscle, the satellite cells, they're gone, and the muscle looks just as fine without satellite cells. So, man, it's not satellite cells. It's very disappointing. I thought there's a lot of cool things we can learn. The other thing is, for this audience, maybe you're not as critical about my work in muscle, but muscle people always wonder, I think you have an innervated muscle, and that's why you don't lose muscle mass. So a bear, for example, other than not being practical to study, also is not interesting to study for me because they actually don't drop their temperature by shivering all winter long. So they lie there, they always shiver. And that shivering obviously keeps your muscle mass, right? If you constantly do this, you keep your muscles. So we denervated the muscle, the leg of hibernating animals. And again, it's not innovation that plays a role in this. So this is now the little bit of signaling talk. I go through this quickly, but because there are some very fascinating findings we had. So if you look at what controls muscle mass from a signaling point of view, the major player and the world leader in this is AKT. That has been, for decades, the major player. And it regulates muscle atrophy by preventing protein degradation through FOXO3A, and then it makes muscle through the mTOR pathway by making hypertrophy, by causing hypertrophy. So what do? You start looking at this from a biased point of view, because by the time I started to do the work, the squirrel genome wasn't annotated yet. It's now, so we can now do a little bit of an unbiased approach and see what else is there. But first we started this unbiased approach. And we saw an increase in FOXO3A. That's what you would expect when you don't have muscle atrophy. And we saw all these other atrophy factors that were not increased, all expected results. The mTOR pathway went up. Again, you would expect that to see in a non-atrophy condition. But what you didn't expect, or what you wouldn't expect, and for sure we didn't, is that the major regulator of this, which should be way up here, was actually downregulated during hibernation. So that was very interesting. So the question then was, what phosphorylates this FOXO3A? It's not AKT. And I have to give full credit to my postdoc and then later on research associate who figured this out. Because I was going into a completely wrong direction for months, and at some point she just figured it out. Because there's one other protein that's known to do that. It's also increasing the P70, that's the mTOR pathway. It's never been studied in muscle before, and it's called serum glucocorticoid-induced kinase. So I'm going to call it SGK, otherwise my tongue is just going to swirl. So what do? You look for increased expression of SGK, and lo and behold, that's what we saw, a whopping increase during hibernation of the total and the phosphorylated SGK. And interestingly enough, when you look after hibernation, it goes down to levels from before. So it's a hibernation-specific event. Just a 30-second taking you back to biology school. There are two type muscle fibers. You have type 1s and type 2 muscle fibers. The type 1 fibers are the ones that make you a marathon runner and cause the endurance exercise muscles. And I really need to change this picture. It's type 2 muscle fiber strength exercise which control making muscle hypertrophy. And I'm showing this to you because you can see in this checkered pattern in the hibernating, all these bright red cells are SGK cells. And if you merge them with a type 2 marker, you see that actually it's the type 2 fibers that specifically upregulate the SGK, which kind of makes sense because the AKT protein, which seems to be similar to SGK, is also in type 2 protein. So that was all very interesting. Obviously, you need to figure out whether this is a hibernation-specific phenomenon, which would be interesting, but not for me. So I'm not a hibernation researcher because I think hibernation is very cool, don't get me wrong. I don't want to try to use it for my patients. So we started a number of mouse experiments. And first we looked at SGK knockout mice. So you start by looking at knockout mice. And the first thing we did, we wanted to see what happens to AKT when you don't have SGK. And not to our surprise, we saw a whopping increase. But we were thinking, so if we have now AKT increase, that should probably compensate for the loss of SGK, and we won't see much. Well, let me say this. Here's the muscle of the SGK knockout mice, and you have seen all the dystrophic muscles before, so that doesn't knock you off your socks. But these muscles are smaller overall, as you can see. Not much, but significantly enough. And if you expose these mice to volunteer treadmill running, you see that they run much less. Interestingly enough, they don't exhaust, so it's not an endurance problem. They keep running, they just don't run as much because they don't have the power. They have the endurance, but they don't have the power to run. And we saw that also when we did some force measurements here, so there's for sure a decrease in this. And then we did another experiment where we kind of wanted to challenge these mice, similar to what we do when we look for regeneration, but when we look for disuse atrophy, we are using this model here, which was developed by French colleagues of mine. I think it's a very elegant model. You take a surgical staple, that's what we use in the surgery room. You take the paw and clip it towards the knee, and thereby you immobilize your shin muscles, the tibialis anterior. I don't know, is there any muscle researcher here? No. Usually I have to defend this model, because the established model is a hind limb suspension model, but that's not disuse, that's gravity. And what happens in space is different than when you wear a cast. So, but I always have to make this case because my more senior colleagues don't like this model. So what happens in a normal mouse? You immobilize them, so you get a bit of a decrease in muscle weight and size. But if you have the SGK mice, you have like an exaggerated response to disuse, kind of proving again that this plays an important role. So the next step was to look at mice that overexpress SGK. And we first repeated this same experiment in this immobilization mice by locally overexpressing the active form. The CA is the constitutively active form of SGK in the muscle. Because if you overexpress it all over the body, it has lots of impacts. It's not a clean experiment. So we just want to see if you just overexpress it in the muscle, what would happen. And you can see that the mice that are injected with the active form of SGK are protected against muscle atrophy. You can see that here it's only the active form that protects against atrophy, which makes it somewhat more interesting. So we just talked about this before. If you look for a drug, it's always better to push something down than upregulating something. But upregulating a constitutively active kinase is a bit less of a headache. So we then looked at transgenic mice of SGK, and they show about a 30 to 40% increase of SGK. So I'm telling this to you for the maybe non-primary scientists, or even the scientists. So my collaborator in Spain was terribly disappointed that he just got so little overexpression of SGK. Because when you do a transgenic mouse, generally you see like 300-fold increase. That's not what happens in physiology. So I was very excited when he told me this, just a little bit of SGK, because that's what we want to do. When I give a drug, I don't want to give 300-fold increase of anything, because I know there's going to be that somewhere. So he was very disappointed because they don't have a phenotype, and he was interested in cancer, cardiomyopathy, and kidney, and they don't have any of this. I actually do think they have ADHD, but he wasn't interested in following up on this. They're very crazy mice. They do have, I think, no, I'm not kidding, they do have ADHD. So, but they don't develop muscle hypertrophy, so, which was okay. My student was disappointed and thought, well, if you have too much SGK, you should get hypertrophy. But they are protected against denervation-induced atrophy. So you denervate these mice and you protect them against atrophy, and that's really what you want. I don't need a super SGK person one day. What I want is a drug that may help protect some of these loss of muscle mass conditions in at least some of these acquired forms of myopathy. So we are adding on to this decades-old paradigm, a new player. I don't think that SGK is more important than AKT, but I do think you need both of them to play a role in maintaining muscle mass. And I think that our hibernating animals really have shown quite strikingly that these animals can teach us something. I mentioned this morning, patients who don't behave the way how we think they should behave, they teach us something too. We can't study them often as much, but it's a very similar concept that the extreme measures of biology are, in my mind, the true gems of what you can see. So the question, is this the only driver protecting hibernation? Well, I wouldn't ask you the question. So we were very interested in mitochondria. who are really the promoters of what we call endurance exercise. So if you look at the marathon runners, their mitochondrial function is top-notch because their mitochondria regulate all of this. And when we looked at type 2 and type 1 fibers, so the type 1 are the mitochondria-rich fibers, you can see that during hibernation, it's a significant increase in these type 1 fibers, and when we actually did mitochondrial staining, you can see that there's a vast increase of mitochondria in hibernating muscle. That's the only tissue, by the way, that increases mitochondria. So my collaborator, who was interested in the eyes, was always looking at was interested in mitochondria too, but the mitochondrial concentration in the eye, as well as any other organ we looked at, goes down. It's just the muscle. Think about how fascinating this is. If you look, this is the most tightly, well-controlled biological phenomenon, I think, hibernation, that we know of, other than humans, probably. So then we looked at the molecular pathways, and then do this quickly so we can move on to the third. So there's an increase in all sorts of proteins and genes that promote the endurance exercise pathway. It's AMPK, PGC1 alpha, the sirtuins, which people know probably from the literature, the red wines, which contains the sirtuins. We should all drink red wine, not just because of cholesterol, but because of sirtuins. But the fascinating result of that was that up till then, the literature has suggested that you either become a marathon runner or you become Arnold Schwarzenegger, also from a signaling point of view. If the marathon running endurance exercise, AMPK-mediated pathway is active, it always suppresses the AKT pathway, and they cannot coexist. At least that's what people thought in their models of studying rats and mice. So obviously, our animals tell us something different. And so how can these pathways be active in parallel? So there was a very interesting paper that came out before we started our studies that YY1, which is a transcriptional factor, can link PGC1 alpha and mTOR, which are these two pathways, together. And you see a walking increase of YY1 in this. So we haven't, so I just told you that I'm about to get the squirrels to Canada, which is difficult. Canadians think of squirrels as pets. Do you know that? I can't get my squirrels here because they think it's a pet. But I think I've convinced somebody to let them in as a research animal. But anyway, I got a grant now to now manipulate all these genes. Now I can manipulate all these genes, but that's my next topic I'm going to talk about, with sorts of cool genetic editing technologies to really figure out what is the major driver of some of these phenomena. And I tell you, my hypothesis is that they all are equally important. I don't think that there's one major driver. So Bruce Spiegelman, who is a mitochondrial researcher, was very excited about this. And we tried to identify in the plasma what is setting off all of these pathways. Because I do think there's some major regulator that sets off a cascade of hundreds and hundreds of different pathways. We haven't found anything yet. So what is hibernation? From a splashing point of view, not too scientific. It's just coordinating endurance and resistance exercise. And I think YY1 probably plays a role in this, and we're going to study this as we move forward. And now for the last, I have 15 minutes, it's good, because I need some time to discuss it with you. It's the lawyer here. There was a lawyer supposed to come here who I wanted to talk to. Anyway. So there's a new era on the horizon, and I call this interventional genomics. And who in this audience, so that's, I apologize to the handful of people who heard this already. You can see that I did a similar good job reminding me now. Who in the audience has heard about CRISPR? So, it's kind of weird to see the same face saying the same story, but I do it for the ones who are not here. So, when I moved to Toronto, the first paper about CRISPR was published. And as you can imagine, when you move to a new city and a new hospital, there are lots of things you have to do. And one of the things which I didn't do was reading this paper, so I made it on my desk, stayed there until the next paper came out in Science in March, and made it also on my desk. And I wanted to read during vacation, but I realized, for the ones who know this, it's a very different language, right? So you have to learn a different language of science. So I didn't read it then. And then that paper was published. It's not actually a paper, but it's just the Daily Telegraph in the UK, who published this as the new way that is going to revolutionize medicine as we know it. And within a day, and that's really true, I had 40 emails from patients who were asking me, can you do this? Does that help my kid? So then I got the necessary kick into the butt. I needed to finally read this, and I became fascinated by this. And so, I don't know whether there's some trainings here. You can change gears if you want. You just have to believe in it, and that's what I did. My entire lab is, I have one person who's going to do the squirrel work, but the rest of my lab is doing CRISPR work now. So we know about this system as a mechanism that actually evolution has put in place millions of years ago for bacteria to protect themselves from getting attacked by viruses. And they do this by cutting the genome of the virus and thereby killing the virus. So Jennifer Doudna, who in my mind is the pioneer of this, people are in court arguing about patents, who is the real leader of this. If they don't stop, they won't get the Nobel Prize for this, which they should. But she figured out that what this bacteria can do to this virus, you can do anywhere in the genome. And that's what it really is. You can do this anywhere in the genome. So what can you do? You can just cut out a double-strand piece of DNA, and that is what you do when you want to create a knockout mouse model. Now, since there are scientists in this group, who has ever tried to make a double or triple knockout mouse? Nobody. You. Painful, long.
We never got it.
Oh, you never even got it. That's even better. Like that. Because you can use that technology now to. It has been shown to induce 4 knockout genes at the same time, and it takes 5 months to do that. So I did once a triple knockout mouse in Iowa City. I started breeding this mouse when I arrived, and by the time I left, it was done, and somebody else studied it because it took 3 years to get a triple knockout mouse. So now you can do this, and you can do it in any model. Chinese people have just done a monkey CRISPR knockout. That's why I was asking for the lawyer who was here, because there's some ethical implications of this work. But you can also cut out a piece of DNA and then supplement it with a repair template and repair any kind of mutation you are potentially interested in. And then you can increase or decrease the expression of endogenous genes. So we all know that there are some diseases that benefit from upregulating or downregulating certain genes. So if I'm, for example, thinking about SGK as a treatment, rather than going through a high throughput screen, we are trying to upregulate this with CRISPR in a mouse now, the endogenous gene, to see what it does. So just to give you an example. But I'm going to show you now a few slides about data. We are working on 5 different genetic disorders. It's a proof of concept to tackle every possible disease type mutation we can think of. So I'm showing you data from Duchenne muscular dystrophy, because these are our most complete data. We have done this for achondroplasia. We've done this for Niemann-Pick type C, which is a compound heterozygous mutation. The other one is a gain-of-function dominant mutation. And we have almost successfully done it. I mean, we have done it, we need to repeat it one more time, for chromosomal duplication, including MECP2. So we're moving 300 kb. That's about, you can probably remove up to 500 kb, just lots of DNA if you think about it. So I showed this to you before, we don't have to go through this. So we got muscle cells from a collaborator in Paris of a patient who has a point mutation in the dystrophin gene right here. We designed the guides for the CRISPR system to specifically target this area. At the same time, here in purple, we designed the repair template to repair this mutation. You can see it's a little bit bigger, but you do that in order to make it more stable so it's not kind of destroyed by the cell afterwards. And here are our results. So if you just give the guide 3 and 4, you don't see anything, which you don't expect to see anything. If you just give the repair template, you wouldn't expect to see anything. And if you give the guides plus the repair oligo, the peak indicates you got corrected mutations. And our efficiency of doing this is about 25 to 30%, which is quite high if you think about it. It's much higher than what has been reported so far, without even using any drugs that push the system to repair. So we are using now different drugs to push the system towards repairing. The other approach we took is an opportunity to kind of, rather than treating one mutation of a patient, to think about something which would treat all patients with Duchenne muscular dystrophy. And what has been known is that a protein called utrophin, or a gene called utrophin, is like the little sister of dystrophin, and if you upregulate that gene, it can compensate for the loss of dystrophin. So if you would put utrophin here, this complex which disappeared here is going to go back. So we use, like I told you, you can use the system to change the actual expression of genes. So we use the system to upregulate the expression of utrophin, and that's what we got here. You see that guide 3, 6, and 7, as well as 1, 2, 3, and 4 for the different promoter regions lead to quite robust upregulation of utrophin. It's about 5-fold increase of.
It has detected that a minimum number of lines are connected to this conference. Do you want to continue your meeting? Press 1 to confirm. If there is no response.
I push 1. And then I just show you here the Western blot of beta-dystroglycan, which is the protein of this complex. So you see, you give utrophin back, you restore the complex, and this week we started to inject mice with a vector called AB9. So that is the current state-of-the-art vector to use for any kind of gene delivery. It's being clinically used in trials for kids with spinal muscular atrophy, with myotubular myopathy soon, and in children with Pompe disease. So as far as we know, it's very safe to use. So we are not hopefully experiencing the same terrible things we experienced 12, 15 years ago with children who got gene therapy and died, and really paralyzed the entire field for up till recently. And then I want to end with this story here. So this is Gabriel, who is a boy who has DMD, Duchenne muscular dystrophy, who I met when he was 4 years old. And I will tell you that he is a very close, the parents are very close friends of mine. So I have a personal connection to this case. They live in London, England. They started a charity, and I'm their scientific advisor for the last 8 years, but more importantly, they're actually really close friends. So I have a personal connection to this case. So this is the composition of the exons of the dystrophin gene, 1 to 79, and what he has is he has a duplication of 18 to 30, like a fairly big chunk of the dystrophin gene that's duplicated. And that's him today. He's 13. So you see he lost ambulation. And although I love this video because he's a terrific kid, making the best out of his life, but he barely can walk. So I told you before that the most straightforward method of CRISPR is just removing DNA, cutting out DNA. So that's what we did. So we designed the CRISPR guides to remove this duplication and restore the exon. We are in the process of analyzing all this, how efficient. But my question to you now is, and that's how we can end, is what are you going to do next? And I literally have not been sleeping for the last 10 days because it's been keeping me up. Because what are we going to do? I will be able to have this experiment finished in probably 3 to 4 months. And if there's nothing totally unexpected going to happen, it's got to work. And then what? So I think that's what's pushing the boundaries means. And I ask each one of you, whether you're a scientist or whether you're a clinician, to walk, not with me, but with anybody who's trying to push the boundaries, because I think we have to do it. There are ways how we have to tackle this question and what to do, and we have to work with regulatory units, because we want to think about therapies like this. We want to think about therapies for rare diseases where you don't have a mouse model, where you don't have a cohort of 10 patients. There's no other patient in the world who has that duplication, and there are many, many different disorders like that. We have to start getting out of our comfort zone and challenge the paradigms how we do this. I don't know how, like I said, this is a 10-day-old problem, but I think we have to do it. So I want to quickly acknowledge some of my collaborators. That's the wonderful woman with the squirrel colony. Leslie Lyman is a very famous muscle researcher who got me into this because she studies pythons. They also hibernate and they have some interesting things. That was my group at Hopkins. These are my hibernation stars. And that's my new group, and that's my CRISPR star, who is my graduate student who also doesn't sleep much because she's working so hard. And I take questions. Thank you. Somebody has to ask a question. Yes.
Thanks very much. That was a great talk. Your work on SGK kinase, you worked on in mice. Does that translate into humans at all? For example, in sarcopenia, do people have lower levels of that gene or anything like that? Have you looked at any studies or human translation of that?
No, I pretty much haven't, and that's mainly because not all of my resources, I don't have enough people to just do this, so I haven't. It's a very good point. We haven't really done any follow-up work on this other than creating more mouse models with tissue-specific either knockout or overexpression of it. They are just becoming available, so we're just getting back into it.
I know a lot of, sounds like you're working really hard at the DMD. I know Joanne talks a lot about, getting retina diseases handled with these types of unique precision types of techniques, individualized techniques. Where do you see this going? Do you think this is all going to be centered in just little centers, like sick kids or whatever? Is this going to end up being dispersed amongst other? How do you see that whole area developing?
So, I mentioned this morning, I used the term, I'm not sure that I want to do this right now or soon, although the window for this boy I'm talking about is closing. Because in another 4 or 5 years, there won't be any muscles left to treat with anything. So I'm going to have to think about this. Where do I see this going? We need to obviously challenge the paradigm, yet prevent what I called this morning the Wild West situation, that anybody is going to say, because it's not even expensive, right? You know how much it costs to do this? Probably $50,000 to $100,000. If I find a company who has AAV9 and I tell them, make these drugs, it costs nothing. If you think about the grand scheme of things. So, okay, here's my dream. My dream is that we are finding a way to regulate this in a way that it's still going to be open to lots and lots of these patients. And that this is not just going to happen at sick kids, but that this will happen everywhere. So I have another 20 years to go. I'm 45. I figure, well, I'm not planning to necessarily retire at 65, but that's the official retirement age, right? I think by the time we go in retirement, you'll do this too. Unless some big unforeseen catastrophes come. No, this will be, we'll be doing these treatments. But we need to start thinking outside the box. And look, if I would know how to do this and had all the solutions, I would have cut my talk short at other places I have presented them to you. I don't know either, but what I do know is that you have to start thinking about it. And you have to bring people together. I have an ethics master student now who I charge to study this from an ethical point of view, with an open-minded person. I know there are probably 50% at least people in here who think I'm crazy. And I respect that. But that's not how you, it's not how you move things forward. And again, I want the ones who think I'm crazy, I'm very thoughtful about this. When I had this discussion with the mother on Friday before I came here, it was a 3-hour discussion we had. She's such a close friend that I can have this type of discussion with her to say, I don't know what we're going to do. And we talked about lots of different things. At the end, she said, I'm looking forward to having this discussion with you in 4 months. I said, you really? I'm not so sure. I said, no. And then we'll make a movie out of it. Would you like to be played by Harrison Ford? I like to be played by Angelina Jolie, so that was the discussion. But I hope that will not know. This should not be central. How about the scientists? Okay. Maybe I give you some stupid comments too.
Are there, I mean, in development, at least, the first tests are always toxicity. I don't know very much about CRISPR. Are there off-target effects like what you'd have with small molecules? I mean, that you can test with by either with screening or antibodies. You can do modeling and see what other things are fine. But are there any off-target effects?
So the one side effect of this technique potentially is that although you design your guides to just cut at that specific area of the genome, there is a computer analysis that occurs that gives you a possible risk of cutting somewhere else, and these are your off-target mutations. So what you do is you design these guides, and then you get a list of up to 50, 60 genes that by computer analysis have a risk of less than 1% to have an off-target mutation, and you have to go through those. So generally what people do in the field right now, they sequence 20 of these genes, and none of us has found off-target mutations. Now I tell you, for this boy, I sequenced the entire genome up and down before I even entertain a discussion of how can I move this forward, which is not going to be feasible. But just to give you an idea, the off-target mutations are theoretically there, but over these 2 years, the technology has been refined to such a degree that it doesn't seem to appear, at least in cells and in live mice.
Could you address that just by doing RNA-seq? Just putting it into and doing RNA-seq?
Absolutely.
Sorry, I just asked if you could address it by doing RNA-seq and then you'd see if there was anything.
Yes, people have done this. I'm not going to do this for all of our patient cells because it doesn't show any abnormalities. I'm going to do it for that boy.
In terms of the CRISPR-Cas repair you were just talking about at the end, what fraction of cells would have to be repaired that way to get back to normal-ish type of? Is there any way to know that?
It's a fantastic question. Thank you for asking this question. So I showed you in my disclosure at the beginning that I'm working with a company to get a systemic gene therapy trial for microdystrophin off the ground in hopefully a year. One of the questions, there are 4 clinicians, I'm playing the clinician role more so than the scientist role on that committee. The one question we ask ourselves, what's success? How do we define success? How much percentage do we have to give back? We know from patients with Becker muscular dystrophy that about 20%, 25% of dystrophin expression seems to be enough to not be wheelchair bound. Still have issues, obviously, but not to be wheelchair bound. So the answer to your question is specifically for this, there's no real target. You kind of have to define a target. I think the target should be 50%. But that is one of the issues of, okay, here I have my AAV9 CRISPR in my hand, in my syringe. What's the dosage? How am I going to look for effective treatment? All of these questions, there's a reason why I'm not sleeping. It's a lot of questions. But your question is unfortunately not a straightforward answer. That's why I'm always jealous about how Dietz, not because he's so successful, but he studies the disease, which you can test easily by doing an ultrasound, checking whether the size of the aorta is larger or bigger, and then you know whether something works. It doesn't work like that initially.
So you have shown that for mutation repair, knockdown, CRISPR can work. Do you think it could be used for the deletion?
If it's not too big, it's not going to work for big deletions. So there's a limit of how much you can cut and how much you can put back at the moment.
Yeah.
So the big deletions are not going to work unless you cut out pieces of the deleted region to create like an exon skipping strategy. So you can cut out a piece of a deletion and induce an out of frame to an in frame deletion. That's what a lot of people are doing right now, actually.
Just a funny question, I'm not a scientist, but that...
Are you the lawyer?
Not the lawyer either.
He's not here.
Just a weird possibility, the duplication of that segment of DNA, could it possibly be an adaptation, or whatever you would say, for some lethal mutation that has enabled the organism to live? And if you get rid of that duplicated DNA, is it allowing for something else in the genome that's gone wrong? Is that how it came to be?
It's a great question. In this case, I would say no. Because evolution has created 79 exons of the dystrophin gene, so having that duplicated causes a disease. And taking that away would be the way to think about fixing it. I wouldn't be worried in that case. But I think it's a very interesting question, which may be true for other conditions. So I think it's a very good question. I don't know what you are, but if you're not a scientist, but that's a good question.